variation, and randomness. The genesis of the confusing idea that variation is random, and the roots of the unfounded conclusion that variation does not play a creative role in natural selection, can be traced to the modern synthesis and to well-meaning but misguided efforts to defend natural selection against creationist attacks, which held that supernatural design, goal-seeking, and intelligence play creative roles. In the light of well-established principles of sexual reproduction, we can easily clear up some of this confusion by distinguishing between two types of variation. The first type is the variation that shuffles segments of the parental DNA sequences during sexual reproduction. This shuffling arises from essentially random segregation of parental chromosomes during the meiotic production of sex cells, as well as the largely random recombination events between those parental chromosomes, which also take place during the production of sex cells. Another source of randomness occurs at the moment of fertilization, because only one of numerous possible sperm cells will fertilize only one of numerous possible egg cells. This first type of variation among individual offspring, random shuffling of genomic sequences, and random fertilization is properly viewed as random variation because the probability of inheritance of any given DNA sequence segment is a function of mathematically calculable probability and chance. This is a blind, undirected natural process that is unrelated to the potential utility or selective value of the possible combinations of genetic determinants that may result. But there is also a second concept of variation that arose at a time when heredity was attributed to units known as genes and alleles, but before their molecular nature were understood. Changes in these genes, resulting in new alleles, were generally described as mutations, and mutations were thought to be random events that are unrelated to the needs of the individual. This second type of variation must be distinguished from the random shuffling of pre-existing DNA sequences during sexual reproduction, because this type of variation includes modifications, additions, and/or deletions in the pool of available genomic DNA sequences. Unlike the first type of variation, changes in the gene pool will potentially apply to several subsequent generations rather than one. They are a source of novelty that is essentially different than random shuffling during sexual reproduction. Today, instead of using the term mutation to describe this second type of variation, we can more accurately describe the range of possible sequence variations collectively as modification, addition, or deletion of genomic DNA sequence elements.